Metabolic pathways come in a variety of forms. You will see often a blend of all of these, okay? So sometimes it's only one, often it's kind of a mix of them. We have linear pathways, so this would be like glycolysis, where you have molecule A being acted on by an enzyme to get molecule B, which is acted on by an enzyme to get molecule C, and so on, D, E, F, so on. We have cyclic pathways, so we're turning constantly to produce molecules with products, and we're inputting them, churning them constantly, like the Krebs cycle. And you have divergent and convergent ones, where you can maybe start with one molecule and then it branches into similar yet slightly different, or you go from branched down to one. This is an example of amino acid synthesis. But again, it's often much more complicated and a kind of blend of all of these, where they kind of feed into each other. Okay, so our next section is on how do we actually get energy and use it. Cells manage their energy in the form of chemical reactions that can change molecules. So they're constantly regulating, trying to find sugars to eat, to get ATP, to build things, kind of like a project manager. Some reactions require energy to proceed. So some you have to input ATP, and others actually you net ATP because they release energy. Exergonic reactions are chemical reactions that will release energy. Often they come with heat being released, but they also come with ATP. So these ones would be where you're probably breaking a bond or breaking up a large molecule and harvesting energy out from it. Endergonic reactions are chemical reactions that you have to put energy in order for the reaction to proceed. Often in the body or in microbes, you will see that exergonic and endergonic reactions are coupled together. So maybe you're going to have exergonic where you're harvesting energy and then you're immediately putting that energy into an endergonic reaction. This is the most efficient way when you can kind of pair them together. Energy cannot be created. So energy cannot be created or destroyed. These are one of the key laws of physics. So it can't be created. It's not like, hey, we're making ATP, but we're just harvesting it or trapping it or storing it for later. Some molecules will readily give electrons, others will readily receive electrons, and this is known as oxidation and reduction. Oxidation is when molecules lose electrons or they donate electrons. Reduction is when molecules receive or take the electrons. Often these are put together into a redox or oxidation reduction reaction. These are reactions where electrons are transferred between molecules. Okay, so just a little review here. I know it's kind of Chem 1 here, but this is oil rig is typically what I use to remember it. So this is oxidation is losing, reduction is gaining. So if we look at table salt here or sodium chloride, the sodium is going to be giving up its electron here in gray. So it's going to be reduction is gaining. Okay, so if we come down here, it's giving it up, oxidation down here. Okay, so we see how the gray has been transferred. Electron carriers are often used within the cell to strategically shuffle energy from place to place or store it. Okay, so these are a lot like, I don't know, public transit, where they're a bus or something, and these molecules just shuffle the electrons from one molecule to the other and back and forth and back and forth. in the form of energy. Okay, so NAD, FAD are two key molecular shuttles that can accept and release electrons to facilitate the transfer of energy. So in catabolism, electrons are going to be extracted, and then these shuttles can carry them until they reach their final acceptor. That final acceptor in aerobic metabolism is going to be oxygen. So aerobic, think aerobes, air, oxygen. Okay, so their acceptor is going to be oxygen. If it's an anaerobe, it will be some other inorganic or organic compound. But if it is aerobic, it's going to be oxygen. Okay, so one of the examples of the molecular shuttle is NAD, where you can see how the electrons are being transferred between oxidized and reduced NAD. Okay, so they're just sharing these electrons between the two, and then if they get to the final acceptor, then they can pass it off. Okay. ATP, or adenosine triphosphate, though, is seen as metabolic money. You may hear it as the energy currency of the cell. It can be earned, it can be banked for later, it can be saved, you can spend it, you can swap it or exchange it, but this is the thing that keeps all the metabolism moving forward. ATP provides the connection between energy yielding catabolism, where we're breaking things down and trying to harvest the energy, and anabolism that requires energy to be put in. ATP, adenosine triphosphate, as you can see, has adenosine or adenine up here with a ribose sugar. This should look familiar to the base pairs that we see in DNA with the ribose sugar down here. Triphosphate, or 3 phosphates, you can see 1, 2, 3. And this one would be when it's carrying the energy. And then if we need to, we can break off one of the phosphates, have adenosine diphosphate, and then when we're ready to store energy on it again, then we can put another phosphate back on. Okay. To phosphorylate or add that phosphate in is phosphorylation, you may hear sometimes. So if we're going to phosphorylate ADP, then we're going to have ATP. ATP is constantly being used and replenished. It's an ongoing cycle of cashing out, storing up, cashing out, storing up. Okay. Energy released from ATP drives biosynthesis. So we need it, for example, this is the first step of glycolysis, where you start input glucose and you're trying to get glucose 6-phosphate. ATP is put in to help drive the reaction forward. As a side note here, you'll notice hexokinase. This is a key enzyme that is needed to expedite the reaction. It has that ACE ending on it. ATP can be replenished by adding the phosphate back on. So we have ADP, you stick the phosphate back on and you're back in business with ATP to spend. All right, next time we'll go further into metabolism to finish up chapter 7.